Hello students, I hope all of you are doing fine. Now today we are going to see CAM pathway. Okay, CAM pathway is called as CAM stands for crassulacean acid metabolism. CAM stands for crassulacean acid metabolism. Now in which kind of plants do we see this? This CAM pathway is seen in plants that are growing in dry and hot habitats. Okay. Now, why did this CAM pathway evolve? That's the question. That's the first question. Why did this CAM pathway evolve? Now, this CAM pathway, the reason for evolution of this CAM pathway is now it is a mechanism for conserving water. Okay, it's a mechanism for conserving water. I'll tell you, we'll understand how it is conserving water in a short while from now. Okay, but remember, it's a water conserving mechanism. Okay, now, okay, let us proceed and let us see what all. Now, it is in which kind of plants do we find it? It is usually found in xerophytic plants like cacti and succulent plants. Okay, and why is it called CAM? Why is it called as crassulacean acid metabolism pathway? It is called as CAM pathway because it was first found in a family called as Crassulaceae. Okay, it was first found in a family called Crassulaceae, and it stores acid. Okay, it stores acid, which is usually malic acid. Okay, it is malic acid. It is malic acid, so that is the reason it is also called as crassulacean acid metabolism pathway. Okay, now let us proceed further. Okay, let us proceed further. Now, as I've already told you, it is found in the succulent xerophytes. Then the primary acceptor of carbon here in this pathway, the primary, same like our C4 pathway. Okay, here the primary acceptor of carbon, okay, here also a primary acceptor of carbon dioxide, atmospheric carbon dioxide, here also is going to be our phosphoenol pyruvate. Okay, it's going to be PEP, and I assume that you know PEP. PEP stands for phosphoenol pyruvate. Okay, it is phosphoenol pyruvate. Now, so this is the primary primary acceptor of carbon dioxide. Now, what is the first formed product in CAM pathway? Okay, so the first formed product in CAM pathway is oxaloacetic acid. It's a 4-carbon acid. Okay, the first formed compound is oxaloacetic acid, and how many carbon acid is oxaloacetic acid? It is a 4-carbon acid. Okay, now what is the significance of this CAM pathway? As I've told you, they conserve water, correct? Now what happens is, in these CAM plants, okay, the stomata that is present is called as the scotoactive stomata. So what is scotoactive stomata? Stomata that are active during the night time or stomata which open during the night time are called as scotoactive stomata. And what are the stomata that are opening during the daytime called as? They are called as the photoactive stomata. So in these plants, the stomata are open during when? In these plants, the stomata are open during the night time. Okay. Now, by this you can understand, when the stomata is opening during the night time there is no sunlight. When there is no sunlight, active transpiration is very, very less. So when the active transpiration is less and humidity is also high sometimes at the night, so when the stomata is opening at the night, okay, there is no water loss to the plant. Water loss to the plant is not there. Just there is carbon dioxide entry into the plant, okay. And during the daytime, the stomata are closed. When the stomata are closed during the daytime, there is no chance for the plant to lose water during the daytime. So stomata open during the night time, and because at night transpiration is almost less or not existing, so plant can save water. Okay, next one. One important difference between the C4 pathway and CAM pathway is C4 pathway, the carboxylation and decarboxylation steps Okay, we'll see this. What is carboxylation step and what is decarboxylation step? This carboxylation step and decarboxylation step these two steps are separated by time. Okay, the carboxylation step and the decarboxylation step, these two steps are separated by time. Okay, they are separated temporarily. Okay, in terms of time they are separated, so temporal separation is present. Which one? Carboxylation and decarboxylation steps. Cran's anatomy is not there. Okay, so there is nothing like Cran's anatomy here. Okay, there's nothing like Cran's anatomy like how we have in our C4 plants. Okay, Cran's anatomy is not there, so absent. Cran's anatomy is absent, doesn't have Cran's anatomy. Okay, so and these reactions, that is the all the reactions, whatever carboxylation, decarboxylation steps, everything is taking place, all of them will take place in the mesophyll cells only. All of them will be taking place in the mesophyll cells only. Okay, but in the case of C4 plants, we have seen the involvement of mesophyll cells as well as the bundle sheath cells, correct? So, but here we have the involvement of mesophyll cells alone. Okay, now let us see the mechanism or how it actually functions. Okay, now first we are going to divide it into day and night. Okay, so we will divide it into what and what? We will divide it into day and night. Okay, so this is during the night time, and this is during the daytime. Okay, and this is during the night time. So during the night time, what is going to happen is phosphoenol pyruvate. Okay, yeah, one more before going to phosphoenol pyruvate, we'll see these are the epidermal cells. Okay, epidermal cells compactly arranged, and stomata are open during the night time. This is what I told you. Stomata are open during the night time. When stomata are open during the night time, what is going to enter inside? Carbon dioxide. And this carbon dioxide that's entering inside will obviously combine with some amount of water, and it's going to form bicarbonate. Okay, HCO3 minus. Now this bicarbonate, okay, this bicarbonate is going to combine with phosphoenol pyruvate. Phosphoenol pyruvate is a 3-carbon compound. This phosphoenol pyruvate, okay, is going to combine with bicarbonate in the presence of the enzyme phosphoenol pyruvate carboxylase. In short, this enzyme is written as pepcase. Okay, this enzyme in short is written as pepcase, which is phosphoenol pyruvate carboxylase, and it is going to give rise to a 4-carbon acid, which is called as the oxaloacetic acid. This is a 4-carbon acid. Okay, now this 4-carbon oxaloacetic acid, undergoes reduction. Okay, undergoes reduction. So NADP So oxaloacetic acid is just undergoing reduction here. Okay, and it is forming malic acid. Okay, it's forming what acid? Malic acid. Now this malic acid is also how many carbon acid? This malic acid is also a 4-carbon acid. Now this malic acid that is formed here, okay, actually it is malate. Okay, now this malate that is formed here enters inside the vacuole. Okay, if you're speaking in terms of very proper biochemistry, it's actually malate that is formed. That malate goes inside the vacuole. This is the vacuole, and here it is going to get converted into malic acid. Okay, we have some pumps here. Okay, these pumps are going to transport this malate into the vacuole. So this is the vacuole. Okay, this is the vacuole. So this is the vacuole. So this malic acid is stored in the vacuole. So this step is called as the acidification. This is called as what? Acidification. So it is getting acidified here, so acidification. So here one more important thing is carbon fixation has taken place here. Okay, so it is carboxylation. Now at night time what has happened? The carbon dioxide got fixed, it is taken up and it is converted into 4-carbon acid and finally stored as malic acid. Now during the daytime what is going to happen? During the daytime this malic acid which is in the vacuole, okay, during the daytime this malic acid which is in the vacuole, this malic acid comes out. This malic acid comes out as malate, okay, and this malate undergoes oxidative decarboxylation, okay, undergoes oxidative decarboxylation, okay, it undergoes oxidative decarboxylation. I'm just writing it as OC, which stands for oxidative decarboxylation, okay, oxidative decarboxylation, okay, ODCL, right, oxidative decarboxylation. So when oxidative decarboxylation is happening, okay, we know malate is 4-carbon, okay, so when decarboxylation is taking place, obviously there will be release of carbon dioxide, correct? Now once carbon dioxide is released, this 4-carbon compound gets converted into a 3-carbon compound which is called as the pyruvate. Now this pyruvate, okay, gets converted into phosphoenol pyruvate, okay, in the presence of by using ATP, which AMP means ATP equal to ATP which is energy which is equivalent to 2 ATP molecules is used here, okay, and in the presence of the enzyme phosphoenol pyruvate dikinase, okay, dikinase, in short I'll be writing it as PPDK, phosphoenol pyruvate dikinase is the enzyme, okay. So this is this one. Next we'll end this phosphoenol pyruvate fate here, okay. Now let us come back here to this carbon dioxide. Now this carbon dioxide is going to enter into the chloroplast, okay, so I'll be drawing the chloroplast here. So this is the chloroplast, okay, double membraned of course, okay, it's double membraned, sorry for this, yeah, it's double membraned. So this inside this carbon dioxide enters inside, and once carbon dioxide is entering inside, whatever Calvin cycle, all the steps that Rubisco fixing, everything will happen inside. Rubisco fixing this carbon dioxide, okay, reduction, regeneration, all those steps will take place and starch formation will occur, okay. So in this process also, okay, how many ATP are required for fixing one carbon dioxide molecule? Same as in the case of C4 pathway, we require 5 ATP, okay, and 2 NADPH, okay, and 2 NADPH. For fixing one carbon dioxide molecule, 5 ATP and 2 NADPH are required, okay. So for fixing one sugar or one hexose, how many are required here? The same number, okay, that is 30 and 30 ATP, 65, 30 ATP and 6 2s are 12 NADPH, okay. So remember this, okay. Now where are the 2 additional ATP used up here for one carbon dioxide? Apart from the Calvin cycle, I assume you know the Calvin cycle energetics, the 2 extra ATP used up are here, okay, here, okay. And what about this one? Whatever NADPH is used here, it is again regained back here, okay. Here when oxidative decarboxylation is taking place, whatever NADP is used up here, this NADP is regained in this step, so this is nullified, neutralized, okay. So this is about the CAM pathway, okay. If any doubts in CAM pathway, you can post it in the comment section. I will try to answer it. Thank you very much.